Cancer is a disease that we've wanted to cure for a long time because it affects so many people. 1/2 of Canadians will develop cancer at some point in their life, and 1/4 of them will die from it. These numbers will be even higher in developing countries with less advanced health care. I know firsthand how much of a toll that cancer can take on someone's life because my grandmother was diagnosed with cancer about 13 years ago. At the time, I wasn't old enough to understand how cancer worked, but as I got older, I was curious as to why she had to be hospitalized for so long and why these treatments that we have today don't work that well. In other words, I was curious as to why we didn't have a cure yet. I mean, we had treatments like using surgery to remove tumors, using doses of radiation and chemotherapy drugs, along with take-home cancer meds to kill cancer cells, and even replacing faulty organs using transplants. What was stopping these from being the cure we were looking for? Well, as I started to research these cures to find a way to improve them, one of the biggest things that I noticed was the cost. Chemotherapy, for example, which is a treatment that is very widely used for cancer, can cost up to $7,000. And on the other end of the spectrum, organ transplants can cost $500,000. This can be way too much for a patient to afford. And even with these high costs, these treatments don't work as well as they should because they attack healthy cells along with the cancer cells. So instead of finding ways to improve current treatments, I decided to go into a newer solution altogether. And when I researched, I found that using CRISPR-Cas9 aided by nanotechnology, we could eliminate all of these problems and find the cure to cancer that we were looking for. Like Hannah mentioned in her presentation, CRISPR-Cas9 is a gene editing tool that uses a Cas9 enzyme and guide RNA in order to edit portions of the DNA. This can basically get rid of the mutations that cause cancer in the first place and cure cancer at the genomic level. The mutations that cause cancer happen when the DNA sequence is created incorrectly, which can lead to the proteins that the DNA is used to code to be created incorrectly as well. For example, if there's a mutation in the BRCA genes for breast cancer, then the tumor suppressor proteins will be coded incorrectly, and this leads to the abnormal, malignant growth of cells that causes cancer in the first place. Something to keep in mind is that cancer is a complex disease, which means it relies on multiple genes, things like environmental factors and family history to create the end result, or the phenotype. But since genes play such a huge role, CRISPR-Cas9 will be a very useful tool. As I looked into CRISPR-Cas9, I noticed that one of the biggest problems with it was that we needed to stop off-target effects and make it more precise, and this is where nanotechnology comes in. Nanomaterials are on the nanoscale and are measured in nanometers. And this means they interact with the world differently from macroparticles and can carry out jobs throughout the body by being self-assembling and active. Some of the nanoparticles that I researched include conjugated particles that can be anything from stem cells to gold particles and can be used to bind to a cell-penetrating peptide and the CRISPR-Cas9 system to locate portions of the DNA. Microfabricated needles can work in the same way, except they use an electrostatic attraction to the DNA to locate mutations, and this eliminates the need for a cell-penetrating peptide since they're a needle and can penetrate the cell membrane. And the third one that I researched was a DNA Nano Clue, which is almost like a ball of yarn made out of DNA, and it can be used to encapsulate the CRISPR-Cas9 system. And using the sequences of DNA created by scientists in a lab to create the Nano Clue, it can correspond to the mutation. So I think this has huge potential because using CRISPR-Cas9 and nanotechnology to create a preventative, more targeted way to cure cancer without killing healthy cells, we could stop all these problems that come from the current treatments. I would want to continue researching this field and even experimenting with it in the future, because by using this, no one will have to go through what my grandmother went through ever again. Thank you.